my pleasure to introduce Professor Shadi Dayeh. He's a full professor in the Department of Electrical Computer Engineering at UC San Diego. He is on an award tour right now. Just in this year, 2022, he's published multiple science translational medicine papers. And I'm very enthusiastic about some of the stuff that he's been doing with neurotechnology. And so he's going to be telling us about that today. So with no further ado, I'd say let's let Shadi go ahead and get started.
Right. Good afternoon. Thank you, Todd, for the introduction, for hosting me here. It's good to see you again, see friends and make new ones. So today we're going to talk about brain mapping devices. This field has exploded in the last few years, and a lot of the advances stand on the shoulders of a lot of clinical work that impacted. To preface the talk, I'll remind you of a few applications and state-of-the-art advancements in this area. You were familiar, perhaps everybody here, with deep brain stimulation electrodes that help a lot of patients with movement disorders. These are electrodes that deliver electrical pulses to deep brain structures, in this case to the subthalamic nucleus, to be able to control tremor in Parkinson's patients. Other work aims at bridging the brain to machine, brain-computer interfaces. Some of the work done here at Stanford have allowed patients to use their thoughts to transcribe text. Other researchers use non-penetrating electrodes like surface ECoG, epilepsy-based grids, in order to decode the brain activity correlated with the kinematics of the movements of the mouth, and then use only thoughts to be able to transcribe speech. And even for simple devices like paddle devices that are used epidurally on the surface of the spinal cord for pain management, spatiotemporal stimulation to contacts on these electrodes allow patients with complete paralysis to now stand up and be able to walk with assisted locomotion. So there's a lot of great things that are happening in the field and a lot of things to happen over the next decade or two. What I'd like you to remember by the end of this talk, a few concepts. The first one is a brain telescope. I'm going to present about a device that will allow us to look at a panoramic view of the brain activity over broad brain regions with microscopic details. We're going to look at devices that look like a display screen, like a computer screen, projecting the brain activity directly from the surface of the brain. And then I hope that I will convince you that thin film electronics is the viable path forward to comprehensively understand the brain and modulate brain function. So this is the outline of the talk. First, we'll provide a foundation, a scientific foundation for the work that I'll be talking about today, how we make the electrodes, their properties, what are the design considerations, and how we're able to scale them for large brain coverage with high spatial resolution. I'll describe one experiment in animals as a gold standard experiment, and then I'll present data in human brain mapping. This is in patients undergoing neurosurgical resection, such as epilepsy or brain tumor resection, and a variety of testing schemes. And then I'll discuss future directions. And so understanding the brain means different things for people in different fields, but even within the field of electrophysiology, we can look at the brain at different scales or details. And if we are less invasive on the surface of the scalp and even go slightly deeper under the skull, we're looking at local field potentials. That is the aggregate of activity from global regions within the brain. And as we get closer to neurons, we start looking at multi-unit or single unit activity. And then as we even go one scale deeper to individual neurons, we can look at activity in ion gate channels. So what we want to have is a technology that can bridge the gap across this whole scale. So we work at the trade-off between understanding the bigger picture with large coverage, but not missing the details with very high spatial resolution. And there's no such technology that can address all of these at the same time. Our lab works on different technologies to answer questions across this range. So today I'm going to be talking about thin film electrodes that can be placed on the surface of the brain to record global local field potentials as well as multi-unit activity. We are also developing micro SEEG electrodes to be able to reach deep brain structures, record and stimulate in a human scale devices. If you want to penetrate slightly within the cortex, we have an array of microneedles with a flexible backing, but I will not be covering this today. They were published earlier this year. And we also have a platform that will allow us to penetrate into individual neurons in vivo and be able to record intracellular potentials from hundreds of cells. The standard clinical electrode that's used in mapping brain activity in epilepsy patients is the ECoG grid. This is one version of it, a research grade grid with high channel count, 256 contacts. This is distributed over 6.4 by 6.4 centimeters with 4 millimeter spacing, and each contact is about 1 millimeter in diameter. And it's built on about 0.5 millimeter thick silicone. Now, with this spatial coverage, it's hard to sample the brain activity at the resolution that we want to sample. And to be able to increase the number of contacts, we need to shrink the diameters. And as we shrink the diameters, we hit the impedance problem, where the impedance has become large, and therefore the noise from the contacts become too high so that we wouldn't be able to discern the signals at the level that we would like to discern them. And this plot right here shows the noise voltage as a function of frequency in order to bring, what I mentioned on the previous slide to data. The noise voltage is plotted here for different contact diameters going from 1 millimeter all the way down to 20 microns. And we can see that at low frequency, the 1/f noise dominates. We have an increase as a function of 1/d squared in the noise as we go to small contacts. This is with planar platinum, but if we use low impedance materials, such as PDOT, for example, or platinum nanorods, which I will talk about later today, we can overcome this problem because of the large surface area, a lot of absorption sites that minimize the 1/f noise, so that the overall noise, when you scale the contact diameter from 1 millimeter to 20 micrometer, is dominated by the amplifier noise, right? So it's not the contact noise anymore. In addition, when we make thin devices, we place them on the surface of the brain, parasitic capacitances can be large. So if the impedance of the electrode is large, then we would be shorting the input of the amplifier. So we can even reduce the signal that we're measuring. So not only the noise, in some cases, the signal can be degraded. Now here we're looking at a clinical grid placed on the surface of the brain of a patient undergoing neurosurgical resection. And right away, you can see that the grid is floating on the surface of the brain. It's not conforming to the surface, and there needs to be an intervention. In some cases, they cut it in order to have adhesion. And you can see the spatial scale is really large, 1 centimeter between the contacts. Now, this is used typically in procedures for passive mapping or stimulation mapping, where contacts are simulated, and in some cases, the patient is engaged in tasks to see the effects of stimulation on these contacts on speech, memory, motion, etc. Right? So they create maps that will guide the resection or the treatment procedure based on the grid. So at UCSD, we used thin film technology. This is microfabrication techniques used for display screens, for instance, to make devices that have large area coverage and large connectorization to separate the electrode in the sterile medium from the non-sterile medium where we are doing the acquisition electronics. And the version that we're looking at is a 1024 channel cortical grid with 1 millimeter spacing between the contacts. So this covers 3.2 by 3.2 centimeters. And we've also used a connectorization that has been developed in the microprocessor industry to connect to thousands of channels through a complete collaboration between engineering and medicine. And then when we, so I'm going to talk a little bit more about this technology, but first I want to show the difference between our grid and the clinical grid. This is the 1024 grid placed on the surface of the brain, again, a patient undergoing neurosurgical resection. And you can see that the grid is very conformal to the curvature of the surface of the brain. It's also compliant to the brain pulsation. So you have an intimate contact between the electrodes on the grid and the surface of the brain. You have high recording for a longer duration of time. It's also transparent so that you can correlate the recordings with the anatomy underneath or place it in the right region that you want to do the recordings from. So what this boils down to is really the interface, like almost every engineering problem, right? So we call it everything is the interface. And here we have the coupling happening in two main mechanisms. One is capacitive, where you have charge screening between ions in the tissue and electrons in the contact material. And then to increase that coupling, we roughen the surface. So you have a larger surface area for capacitive coupling. There is also the second mechanism is Faradaic interaction, where you have redox reactions happening at the interface. And when you roughen an electrode surface, you create dipoles at sharp contacts, at sharp points, and that lowers the work function, making that interaction or redox reaction having a lower energy barrier to happen. So it's not only the surface area, the electrochemistry becomes even better. And this has been applied in sputtered iridium oxide films, or even in PDOT, which has a volumetric sensing mechanism, because it's a one-dimensional polymer, so ions can penetrate in between these one-dimensional long polymer chains and then interact with the polymer layers. But if we want to design an electrode that has the highest sensitivity, we need to look at what's called a volcano plot from electrochemistry. This is looking at the exchange current at zero bias as a function of metal hydrogen bond or metal ion bond. And if we have a small bonding energy between the ion and the metal, which is the surface of the electrode, that means that ions in tissue will have small residence time on the contact and therefore have less chance to participate in a Faradaic reaction. If that bond energy is high, that means that the metal ion will have a long residence time. There is one Faradaic reaction, but you're blocking that adsorption site. So the sensitivity is low. So you just need a metal with the right metal to ion energy so that the ion can come in, participate in the Faradaic reaction, and clear the adsorption site for other ions to come in and again repeat the process. And that's why platinum is the most efficient catalytic material because of its suitable metal to ion bond that allows it to have a lot of these Faradaic reactions without blocking the adsorption sites. And then to increase the surface of platinum, we were inspired by this work that allows us to do nanoscale structures or high surface area structures with a low temperature process that's suitable to be made on flexible polymer films. So this is essential so that you can respect the thermal budget of the substrate. This process is called de-alloying, where an alloy of silver and the metal that you need for, in this instance, what we're seeing here is gold, and we applied it for platinum. So gold and silver were co-sputtered, and then they were inserted in a hot nitric acid bath where silver can be selectively etched away. And to minimize the surface energy, silver then diffuses to the surface, and this process continues until you get a nanoporous structure. So we optimize this process in order to make platinum nanorods. This is the single contact, and the rods are recessed in the contact below the surface. If we zoom in, we can see a mesh of platinum nanorods made with this process. And here we're looking at a picture of the device, and below a CM image where you see perfusion holes in between the platinum contacts. This is on the right-hand side of the slide for those who are watching by Zoom. And on the lower right, we're seeing an image of the device. It's 10 microns thick. The platinum contacts are usually 30 microns in diameter, and perfusion holes allow perfusion of CSF from underneath the grid so that we have that intimate contact with the surface of the brain. Now we'll have 2 slides on electrochemistry, and then I start talking about electrophysiological measurements. So first, I wanted to show on the top left that this is a highly reproducible process. We're looking at 56 microcontacts here that have a nearly identical impedance spectra. And now we have this for thousands of channels. On the lower left, we're looking at a comparison of the impedances for planar platinum in green platinum nanorods and in red P.PSS. And we see that platinum nanorods have about 1 order of magnitude or more lower impedance than planar platinum. So it's there with the class of the highly electrochemically effective material like P. So this is for recording. Again, you want the low impedance for low noise. For stimulation, you would want to inject sufficient charge density to stimulate the tissue without leading to adverse effects, without damaging the tissue. And to understand that, we used a parametric characterization of the electrode tissue interface. Here we're looking at what may look like a complex picture, but it's really not. This is for bipolar stimulation between 2 contacts. And the interface exists from a simple Randall cell that have a double layer capacitance for capacitive interaction, a Faradaic or redox branch. And then we have a series resistance, which is mostly current crowding at the edges of the contacts, and then the tissue impedance. And we are concerned with the voltage drops across this interface right here within half a nanometer or so from the contact. There, you want your voltages to be low so that you don't cause electrolysis of water to result in oxygen species or to result in hydrogen bubbles. So we can measure the voltage drop across each of these components and then predict which currents can be safe for stimulation. So how do we do that? We start with cyclic voltammetry, in which we expose the electrode to potentials and look at the current surges. Those current surge regimes, for example, on the right would be the regime where we start to have oxidation, when we start to have hydrogen bubbles on the left, and those would be the limits for electrolysis across the electrode tissue interface. And then we start pulsing the electrode with different current pulses and estimating the potential drop across that electrode interface that we talked about, the half nanometer or so, both in benchtop and in vivo settings. And we do parametric studies for different pulse widths, different currents. And we want to make sure that the voltage that's being dropped is below the voltage that we estimated from the cyclic voltammetry. And that will be our current limits, or define what's called charge injection capacity, the total amount of charge that's normalized to the area. So on top, now we see that charge injection capacity for platinum nanorods compared to P. or platinum, again, over 1 order of magnitude higher than the planar contacts. You would also want to test the stability of these devices with millions of cycles, and the plot over here shows that their impedances are stable over a long duration of time. Now, the gold standard for deriving the safe currents in electrical stimulation clinically and in research is the Shannon's equation, which relates the charge density to the charge per phase by an empirical formula. And that formula doesn't account for nonlinearities for current spreading, nonlinearities with area, nonlinearities with the pulse width. So here we did a study both in benchtop and in vivo to be able to estimate the voltage drop across the electrode tissue interface, and then calculate the safe currents that we can inject into the tissue without causing adverse effects. And this shows basically how the model takes into account the nonlinearities, as I mentioned, in terms of pulse width, also the impedance of the device. So the material also is taken into account in this model, and fits the data that we measured fits very well with the model. And we've also applied it to clinical electrodes. On top, we're looking at fits for the voltage as a function of pulse width or current. in benchtop and bottom in vivo. And the model fits also the data from clinical electrodes. So if you're interested, please take a look at this paper in Frontiers in Neuroscience that was published recently. So now we'll talk about the application of this electrode in mapping activity from the human brain. First, we look at the devices that state-of-the-art research devices that our colleagues are publishing. ECoG collaboration looks at polyimide material with platinum contacts or with iridium oxide and other contacts, a few channels, maybe 32 channels. Other colleagues at Osaka University use tiles of contacts to construct over 1,000 channels used in non-human primates. And then a collaboration between Duke University and BlackRock uses liquid crystal polymer in order to record also from the surface of the brain of patients. And this is with a few hundred channels. Our fab is done on large glass plates, relatively large. So these are 7-inch by 7-inch or 18 by 18 centimeters. We're looking at the first generation devices where on top we have the grid. We had at the time the large connector, and this connector is becoming smaller and smaller and eventually wireless, as I will show toward the end of this talk. Today we are at 4,096 channels. They are 1 millimeter spaced, and the contacts that you see here are the larger contacts for stimulation. These are 256 channels at 4 millimeter spacing. The design is tailorable, so you can change the distribution of the contacts based on the area in the brain that you want to record from. So these are different versions in the middle, the 200 micron pitch is for recording from the superior temporal gyrus in language tasks, as I will show a little bit later. And then we have high density rat electrodes showing in the other three figures with the density of up to 100,000 per centimeter squared. So we'll start with mapping in a gold standard experiment where we know that there is organized distribution of cortical columns. This is the whisker barrel experiment. So we place a grid with spacing between the contacts of 150 microns, covering about 4.5 by 4.5 millimeters. And we bring a small tube, do air puffs on individual whiskers. So we want to evoke activity in the sensory motor cortex that corresponds to that individual whisker. So when we look at the data from 1024 channels, you see a localized activity, and there is some delay also in waves or in signals that appear at other channels. Now, if you distribute it spatially, this is for the E3 whisker, and then the E4 whisker, we have a shift in where the activity is mainly concentrated. So we've done this for multiple rats, and we always see the same behavior, that is, the activity is aggregated in certain regions that are within 0.5 millimeter or so. We're looking at the lower left image. This is for the fourth rat, where we see distributions for the different whiskers. And if we do electrical stimulation for the neck, trunk, tail, etc., you can see also similar localized distributions. Now, these whisker barrels used to be stained with VGAL2, and then you can identify them clearly with this staining. So we did that for the same rat, and we overlaid the functional high gamma activity mapped functional units over those that are identified from staining, and we see one-to-one correspondence. So now we can measure basically cortical columns from the surface of the brain without staining in a single shot in one array. Right, so we were encouraged by this result, and we applied it to humans, and I'll show some results from humans a little bit later. Now, in addition to looking at individual potentials, we followed approaches in the field to look at brain waves. This is done in the context of sleep studies, where people looked at sleep spindles for consolidation of memory. And basically what's done here is looking at the gradient of the phase of these signals of a certain period of time, and then deduce the wave pattern in these signals. So here on the right, we're looking at a plot of the brain waves, where we're looking at the spatial gradient of the phase, and in the background, we're looking at the potential distribution. And now you can see that we're accompanied with the potentials, we have waves directed as in a sync source type configuration. And also you can see that there are residual brain waves separate from the function that we are recording. When they collide together, they form what's observed here like a spiral. These are multiple waves in close vicinity. Now, having this high resolution is very important, right? You can see the level of details that we can get with the 1024 channels. And if we undersample from the same data in 16 by 16 or 8 by 8, you start to lose some of these wave patterns with the lower resolution. Now, to take this work to the operating room, we need to first go through the process of packaging and sterilization so that it's compatible with the surgical flow. So we've done this, came up with a way where we can have part of the device to be exposed to the sterile medium and the other part to be encased in sterile casing or bag, as we see right here. This was our first version of the recording electronics based off in-pan amplifiers. Each one of them had 64 channels, so we have 16 of those. Now we're making our own amplifiers with colleagues at UCSD. And first we do some testing in the OR to make sure that the sterilization process was okay, didn't damage the electrode, and then electrodes are placed by neurosurgeons on the surface of the brain, typically for about 20 to 30 minutes during the downtime of the procedure. More recently, we've changed this approach to be compatible with shipping across multiple states and used sterilization trays that are typical for the industry. So this is showing pictures of how this is done, the final packaged device with labeling, which is, of course, what the FDA requires to be able to get clearance for their use. So a lot of devices, as we've seen here in this picture, done by graduate students or postdocs to be able to do these studies. So we'll start looking at the human experiments, and this is done chiefly with two colleagues, one at OHSU, Dr. Ahmad Raslan, and at UCSD, Dr. Sharona Benhaim. And the first measurement that's done during the open craniotomy is defining where's the central sulcus. It's the landmark to define the remainder of the procedure. So here, basically, the electrode is placed over what's called the central sulcus, and an electrical pulse is applied to the peripheral nerves, and that electrical pulse leads to phase reversal across the central sulcus. So in one region, you have a negative first signal response. On the other region, you have positive first response. Now, in the middle picture, we see the grid placed on the surface of the brain of a patient undergoing this type of procedure. The central sulcus has this large blood vessel, but if there's a lesion in that place, there is usually brain reorganization, and that boundary can shift. So we really need to measure that electrically to be able to tell where is the sensory and where is the motor regions. And we've done that. And this picture that you see in the bottom at the lower center is defining the functional boundary between the motor region and the sensory region right here. So compared with, again, undersampled 16 channel, it's very hard to look at the curvilinear nature of that boundary. It's a matter of decision of the surgeon on how to define that boundary at that point. But the grid with the 1 millimeter resolution allows us to look at the curvilinear nature of that boundary. In some cases, patients are awake, so we can engage them in experiments. Here, we placed vibrotactile stimulators on their fingertips, and then we time-locked the excitation with recordings from the surface of the brain, so we can identify individual fingers from high gamma activity on the surface of the brain. And this is an overlaid picture on the lower right of all the fingers. And you can also see here that the responses, this is a sensory experiment, are within the sensory region that we've identified with the first measurement, right? So they cross-correlate. In other cases, we ask patient to perform some tasks, in this case, a hand grasping task, and we can monitor the neurodynamics during or before, during the execution and after the execution at different frequency bands. And going to show a video of how this is done. We usually have hardware to track finger movements or hand movements, and the signals are time-locked to what we are measuring on the surface of the brain. This is during a hand grasping motion. And again, we're looking at the spatial derivative of the face. We start seeing waves bouncing back and forth between the motor region and the sensory region. And this is in a 2D projection. All right, so we start to have potentials first in the motor cortex and then in the sensory cortex. And throughout the execution, there are waves that are bouncing between these two regions. And this is a snapshot across this activity from 85 milliseconds to 300 milliseconds, kind of tracking how the potentials change during this motion. And then taking a line cut across the white labeled line on the top right, we can see the waveforms on the right-hand side of the lower images. There is a lag lead relationship between the sensory and motor cortex that changes across or alternates across the hand grasping motion. So now basically we started looking at the brain activity in time and space in terms of brain waves as opposed to spatial correlates of motion. And so if we used to rely in a BMI tool on potentials that are measured in individual contacts, and if these contacts go bad, then we miss the function. But if you have it as a wave on all contacts, then even if a few contacts go bad, you will be able to still capture that motion or that intended function. Now the grids have perfusion holes as discussed earlier and as shown on the top right. So we can stimulate through the grid, and this is a clinical procedure. It's called Osman probe, in which typically the patient is awake performing a task like counting, and stimulation is delivered to the surface of the brain so that the surgeon would observe there is a slur in the counting. And if there is a slur, then they would say that, okay, this location is correlated with speech, so we should avoid resection there. And typically this is done by removing the grids and then doing stimulation separately. But here what we're showing is that you can do it through the grid, and while you're recording the responses to the stimulation at the same time. And in the future, when we get FDA clearance to stimulate, we'll be able, of course, to do the stimulation from the same grid. So you have one placement for recording and for stimulation and one system. So again, consolidating the surgical flow or the functional mapping flow. Now here we're looking at epileptic discharges. These are the black dots that you are seeing across the grid. The contours are the potentials that accompany these epileptic discharges. And this is an epilepsy patient. We see that the discharges started at the top of the grid and circled all the way down. And then when electrical stimulation is applied, that repeated wave pattern was aborted. We have after discharges in the location where stimulation was delivered. And then there are localized regions we didn't see in this video in which there is still residual epileptic activity. But the point here from this video is that we can now look at epilepsy at a microscale resolution where you can track the node of where epilepsy started and be able to deliver treatment either by stimulation or by resection to a much smaller brain tissue. Right, so here it's showing snapshots of this wave propagation, as I mentioned before. And for those who like to look at waveforms, these are the interictal discharges that were detected and then displayed in the video. And lastly, in terms of brain mapping, this is a high-density grid with 16 by 64 contacts at 200 micron channels or 200 micron spacing, placed on the superior temporal gyrus where language is decoded in the human brain. And the patient is listening to different sounds, particularly phonemes here. And we're looking at responses to phonemes, responses to noise, and identifying the spatial extent of these responses. So here we're looking at the high gamma response as a function of space for the phoneme G. And you can see that the phoneme evokes activity within, again, a millimeter type space across this grid. And when we do this across many trials and across many phonemes, you start seeing a mosaic distribution of where phonemes are decoded on the superior temporal gyrus. Now, we are attributing these to be columns or functional units for the phoneme decoding on the surface of the brain. And this happens across multiple patients and measured again from the surface without having to penetrate into the brain tissue. Implications of this, not only for understanding language, but potentially for developing a high resolution speech prosthesis implant. All right, so we looked at mapping the activity from the surface of the brain. A lot of times in the context of epilepsy, the epileptic focus is below the surface of the brain, below the cortex. So you need deep brain or SEEG type electrodes to reach deep brain structures for mapping. And in the case of other diseases like Parkinson's, as we've before, for stimulation. And so there's another effort basically to make high channel count deep brain electrodes. We first overview state of the art in this area. Again, the LNL-UCSF collaboration looks at polymer-based electrodes that are rolled over medical tubing and implanted in the brain. Almost every development is based on planar technologies that are rolled over biocompatible or medical grade tubing to make the depth electrodes. What we've done is a completely different process that I will introduce in a second. But first, how does a clinical depth electrode look like? This is what this picture is showing us. This is an AdTech electrode, typically has 8 to 16 contacts at the surface. They are a few millimeters spaced from each other. Each contact can be 2 to 3 millimeters in terms of its extent, sometimes larger. The diameter of the electrode is at smallest 0.8 millimeter, at its largest, actually it could be larger than 1.2 millimeter, but for epilepsy monitoring, usually the range is 0.8 to 1.2 millimeters. If we want to record single unit activity, there are some alterations to this electrode where the tip, for example, is open and micro wires are inserted so that one can record from individual cells. And usually this is up to 8 wires. So we use thin film technology. In this case, we're looking at polyimide electrodes to make high channel count mapping depth electrodes. These are inflatable electrodes, as I will show in a second. They have a similar length to the clinical electrodes, so 28 centimeters. So you can imagine how long these electrodes are so that they could be properly handled by a surgeon very quickly in a high pace operating room. Now, if we look closer at the tips of the electrodes, we're looking here at a stainless steel stylet that usually hardens a clinical electrode. In this case, it's hardening a polyimide electrode for insertion into the tissue. This is the top, this is the back. And if we zoom in, we start seeing the micro contacts on the top left. There's an array of holes at the very tip so that we can have better adhesion between the two polymer layers at the surface to prevent ripping. And this is the back of the electrode in which we release sacrificial layers inside so that the electrode becomes inflatable. Now, on the lower left, we're looking at histology in the same rat, but implanted with 2 electrodes. One is a clinical electrode on the left. One is our micro SEEG electrode on the right. You can see that the extent of the damage is much less with the smaller volume. Also, away from the electrode tissue interface itself, there's a lot of neuronal death. These are the blue stained neurons. The green neurons are the ones that are still healthy. But in the vicinity of our electrode, there is a lot of still alive neurons very close. So the extent of the damage, of course, of the clinical electrode is much higher. It's much more invasive than our electrode. Now, this shows the scale, basically, of the contacts on this plot. With the large contact, you do spatial averaging when you record activity, and that's why typically we are looking at local field potentials. But with these microcontacts, one would be able to look at individual neurons and synapses. And so we validated this technology in multiple species, from rats to pigs, non-human primates, and in humans as well. This is an example of using the long electrode in non-human primates and recording at different depths. On this slide, we're looking at a typical montage. Even in real time, you start seeing single units recorded on these microcontacts. And then this is another picture showing the waveforms of these units that we can isolate different depths. And this would allow us basically to look at the different neurophysiology or cell types as we go across the different layers in the brain. Earlier versions of this electrode, we call it laminar, has been tested in humans at Mass General. It was with PDOT-PSS, with Sid Cash's lab. And again, here we are able to discern activity in response to auditory stimuli at different frequencies and observe differences, measure differences across different channels. And lastly, I want to talk about the high density depth electrode. We took our high density planar grid and we also applied it to a clinical depth electrode that we see right here. This is a scale of 250 microns. So we're looking at 31 micron spaced contacts, 1024 channels. We did pilot studies in rats where we stimulate whiskers from different directions. And we started measuring responsive regions that change with where the air is being blown on the whiskers. Now, in the last maybe 10 minutes or so, I'm going to talk about future directions, what's going on currently in the lab and where we see this area is going. So we recently have a grant from the NIH to do a wireless epilepsy monitoring unit. And that's to solve the problem of the burden of wires in the epilepsy monitoring unit, also to reduce the risk of infection for the externalized wires, and also to exploit the high channel count or the high resolution in mapping the activity from either the surface or from within the brain. So here we're looking at how these electrodes would be placed, where after making a craniotomy, as we've seen earlier, the electrode will be placed on the surface of the brain subdurally. And we have a 4096 channel, 1 centimeter wide lead, multi-metallizations, that's rolled around a medical grade tubing and connected to an acquisition electronics plus a transceiver and coils that are placed on the surface of the skull. After closing the craniotomy and doing some bandage on the surface of the brain, there will be a head-worn unit that will take the inductively coupled data from the subcutaneous unit and transmit it wirelessly by Wi-Fi to the control tower. And this is an effort with a number of colleagues, transdisciplinary across, of course, engineering and medicine. Another area that we started looking at is mapping the activity directly from the surface of the spinal cord during spinal cord surgeries. This is called intraoperative neuromonitoring. And the usual way this is done is by stimulating, again, peripheral nerves on the legs and the hands and recording activity by EEG from the surface of the scalp, or applying a high voltage, low current pulses to the scalp or to the brain, and then recording the responses in the muscles by EMG. And this is happening continuously during the surgery. We want to look at the evoked somatosensory potentials directly from the spinal cord and potentially identify what's called the midline, which is the center that separates the right and the left propagating fibers, very much similar to the mapping of the central sulcus we've done in the brain. Now, this hasn't been identified before with high resolution in 2D. What we're looking at in the right-hand side snapshots is the time propagation of the response over the grid. And we're looking for the response to the left median nerve stimulation and Below it, we're looking at the response to the right median nerve stimulation, and they're spatially distinct so that when we overlay them over the grid, over the spine, now we have a 2D map of the midline that separates the two. And what we've observed is that even when clinical electrodes fail to detect any signal, as we see right here, which typically means the surgery needs to be stopped or at least halted until the signals come back in the peripheries of the patient, we're able to still see responses on the grid. So on the left, we're looking at the subdural response to the left median nerve post the resection and right to the right median nerve. While the EEG didn't measure activity, the grid was still able to measure activity directly from the surface of the cord. In some cases, you would want to record all the way through the surgery while doing the resection. So here, 2 weeks before the surgery, we were told about the size of the tumor that's being worked on. And so we designed the electrode to have a flap within the electrode itself so that we can first record everywhere, open the flap, and the surgery proceeded in the opening, as you see in the lower video right here, while the recording resumed throughout the surgery. And lastly, we would want to consolidate the surgical flow in the operating room. This is a picture that will show the spatial distribution of the key stakeholders on the operating room. You have the surgeon on one hand, anesthesiologist, and then the electrophysiologist really in the back of the room. So the information flow between the two is really very inefficient. And a lot of times the electrophysiologist marks on a piece of paper the contacts that are epileptic, for example, or correspond to language, and take that paper to the surgeon to be able to make the clinical decision. Now, my lab is a solid states lab, so we make light emitting diodes and transistors. So here we're looking at gallium nitride LEDs, micro LEDs on polyimide films. This is 15 micron thick. And this is a scale of 3.2 by 3.2 centimeters, the human grade scale. And so what we've done is we've applied the grids, we've applied the LEDs on top of the grids so that we can analyze the data in real time and then display the activity directly from the surface of the brain to inform the surgeon so that they don't have to, basically go through the process of exchanging that information with the electrophysiologist in the back of the room. So this is a picture showing how the grids are integrated with the LEDs. On the right-hand side, you can see the back of the LEDs, which is facing into the page. And then this is the micro contact, which is facing us and would be facing the brain. And then most right is just the other direction where we see the light coming out from the surface of the LED. Below, we're looking at a rodent scale grid with smaller area coverage. And then once we integrated the LEDs on the grids for the rats, we repeated that whisker barrel experiment I introduced earlier. And now for different whiskers, we can see that there are different spatially evoked regions for these whiskers. So now we can basically display these cortical columns directly from the surface of the brain. If we treat the brain with a epileptic-inducing drug like bacuculin, then we can observe these discharges in real time. And this is what you're looking at here. These are epileptic potentials filtered in the frequency range of 10 to 59 hertz. And you can now see them, where they start and how they propagate in the real time scale. On the right-hand side, we're looking only at the interictal discharges that has been processed through a special filtering window to be able to detect only these IIDs. So we hope that with this information, that we shorten the time of the procedure because the surgeon will be informed where every disease or functional tissue is and be able to take better clinical decisions. We have a platform of piezoelectric devices. These are pressure-sensitive devices that you can modify in order to detect shear, as we were seeing on this video right here. And we use that initially to be able to detect slip in objects and close the loop in robotics using only sensed activity from the shear sensors. And now with the capability of measuring the brain waves that's correlated with hand grasping, what we'd like to do is develop a plug and play brain-machine interface where you would need lower number of trials to figure out what's the intended function. So first we want to demonstrate that with the shadow hand, which we got earlier this summer. And then we want to provide the sensory feedback with the pressure sensors that we've seen here. There's some work on the spinal cord stimulation that I didn't go over today, but what we'd like to do is bridge the gap in spinal cord injury between the brain and the spinal cord. And so overall, what we envision this field is going to is personalized brain implants, where you look at non-invasive imaging of the vasculature of the anatomy and potentially some function with MAG and guide the electrode design. And with being able to put 1,000 channels per grid and potential per depth electrode and potentially 4,096 channels, we're looking at least 64,000 channels to 100,000 channels per individual patient, meaning that there are a lot of challenges on the acquisition electronics, transmission, wirelessly or wired, the data handling and display, and then what we make out of this information. So to do that, we have partnered with a number of clinical institutions across the country. We hope to partner with colleagues here at Stanford so that we can really take this work from the lab to the bench side. A few years ago, we were at 32 channels in 2012. By 2019, we were at 1024, and today we are at 4,096. We're trying now to cross the barrier of using the 4,096 channels from the acute domain to the semi-chronic epilepsy domain. And what we hope in the next 10 years or so, we'll be able to do the 100,000 channel grids and depth electrodes in humans chronically. So the things that I hope you will remember from this talk is that I showed you a grid which provides high resolution mapping from the surface of the brain over large areas without losing the resolution. I've shown you that we can display this activity directly from the surface of the brain, similar to our display screens by light from light emitting diodes, and that thin film electrode technology can be indeed a viable path for minimally invasive or less destructive brain-machine interfaces, and as a science tool to understand how the brain works in disease and in normal function. A lot of the data that I've shown comes from my lab and a number of colleagues' lab, particularly Sid Cash and Eric Hallegren, as well as my colleagues Ahmed Raslan and Sharona Ben Hayem, and those who are not named on this picture. And the people who produced this data is, of course, graduate students and postdocs that are shown here on the lower left. In 2019, we started to get NIH support that allowed us to exponentially increase the number of channel counts. So we're grateful for the NIH and the early investment from the NSF and then internal grants at UCSD. And so that's all. I hope you enjoyed learning about these new electrode technologies. You learned a little bit about the material aspects that goes into designing them and what they can measure from the brain. So I'd be happy to take any questions. Thank you.
Any questions in the audience or on Zoom? So a question here. So in order for people on Zoom here. Okay.
Yeah, thank you so much. That was very impressive. Thank you.
Thank you.
I'm wondering, like, I was very curious about the last slide that you were showing when it integrates together light emitting devices and recording pads. How do you perform addressing of your single light emitting device? And was it a particularly like complicated task to combine it with a flexible, semi-transparent system? Because from the images, it looks like all your backend is somewhere further away from the actual recording pads, right? Like, could you maybe explain it a little bit more?
Yeah, sure. So the question is basically how to combine the LEDs with the grids. Is it easy? How to address them? And this is multiplexed addressing of the array, right? So we have a small number of wires to address them. It uses chips that are already being used for running display screens, right? Because display screens run on light emitting diodes. So there is a lot of advancement there. We had to make them on thin substrates so that they can be conformal to the brain or at least not disruptive. So they're done on 15 micron thick polyimide. The grid itself is done with parylene C material, which can conform better to the surface of the brain, but it wouldn't be good enough to tolerate the currents that will drive the LEDs. So we had to switch to polyimide to do that. And we temporarily adhere them on one end, align them prior to the surgery. And in most placements, it's nearly localized on top of the grids. But we know that there is a three-dimensional nature of the surface of the brain, so it's not going to be exactly one-to-one, the projection. Did that answer all the questions? All right. All right. You're welcome.
It was an interesting talk. Now, I have a question about the mechanical property of these chips. How would you overcome the stiffness difference between polyamide and the brain tissues?
There is a very big difference in the Young's modulus that you can compensate by making the material much thinner. So as it goes thin, then it can become more compliant. And it's no more a bulk property that matters because the thinner material would have effectively a lower Young's modulus that can't match that of the brain.
And also one more question. You mentioned about the wireless power supply. So as the number of the channel increases, with the power supply enough with those wireless device?
Yeah. So that's a very good question. It depends, of course, on what's the rate at which you want to operate, the sampling frequency, and higher sampling frequencies will demand more power. For epilepsy monitoring, we're looking at a 2 kilo samples per second sampling rate. And for that rate, we can operate the device with an overall power budget less than 50 milliwatts per centimeter squared. So then it's an acceptable amount of power density so that it wouldn't hurt the tissue, wouldn't raise the temperature enough to hurt the tissue. And it can be powered by a battery. So you don't need a continuous power supply or changing the battery too many times. But if we go to higher sampling rates, then that means the battery needs to be changed more frequently or a larger battery is used. Yeah.
Sure, click it on.
Thanks so much for the talk. Super informative and inspiring.
Great.
I was curious, you said an aside about, I'm also an open EFIS and Intan user, different application completely.
Yeah.
But you mentioned that you're building some of your own chips with some of your own amplifiers. I was wondering if you could sort of expand a little bit on kind of the motivation for that and what you're hoping to get out of modifying the chips.
Yeah, so right now there is really no chips available commercially for thousands of channels. We want 4096, so we're looking at 4 chips to do that. Each one is 1024 channels. We will be using Open EFIS because it's one of the few softwares that's capable of looking at data from thousands of channels. So we have a collaboration with the Allen Institute to adopt many modules from Open EFIS, make it GMP compatible so that we can take it to the FDA for approval for use in the epilepsy monitoring unit.
That's amazing. And then is there a reason, so then you just need to make your own amplifiers to do that many channels or were there, okay, so it's not like a performance issue on the.
No, no, yeah, Intan is really good. One of the best amplifiers that are available there for low noise, wide dynamic range recording from single units all the way to local field potentials that are millivolts. So it's not performance, it's mainly the channel count.
Cool, thanks much.
Yeah, you're welcome.
Thank you for the talk. I have two questions. First, can you go to slide 57?
Sure. Two more. Okay. Here?
Yeah. So in the middle picture, what is the difference between this vision and like what Neuralink is doing now with the multiple distributed electrodes and the wireless receiver?
Okay, for the wireless part only, let me see. I wanted to draw analogy between different things, but what Neuralink overall is doing is invasive imaging of the vasculature to inform where the electrodes will be threaded. We'll be doing it non-invasively. In terms of wireless, I think Neuralink right now is doing batteryless inductive coupled power and data. And we will be doing batteryless inductive power and data in a subscalp or subcutaneous unit that's under the scalp, above the skull. I don't know if in their approach they will be removing part of the skull or not. Very likely they are. The initial phase of the application is for semi-chronic monitoring for less than 1 month or less than 30 days. I think their goal is to do it chronically for a long time. What else? Our grids and depth electrodes have the electronics always integrated on the end above the skull, but not anywhere close to the tissue. I'm not sure how far are their electronics from the tissue. Did that address some of the things that you were wondering about? Or perhaps you could be a little bit also more specific about the differences that you want to know.
No, I think you addressed them. It just the picture looks very similar to the picture I have in mind of what Neuralink is doing. And then I have a second question. What motivated you to look at the gradient of the phase for your activity as opposed to just like raw activity?
To be able to look at basically time delay, right? How the waves propagate. And you can only do that with looking at the phase of the waves. Yeah.
Exactly was the signal you're trying to extract looking at the time delay?
We wanted to look at, okay, the potential appears in one cortical column, but then is communicated to different brain regions. So we wanted to track how the potential is moving and across which path. And you can only do that by looking at the wave properties.
Is it working now? Okay. Thank you again. And one more question. So you mentioned that your electrodes adhere conformally to the brain surface. Does it still require some additional surgery or placing of like stability anchors to remain in the exact location and position to ensure like semi-chronical operation or chronical operation? I would imagine maybe FDA approval might request this, right? So that it doesn't move and the patient does not require the surgery immediately.
Yeah, usually when it's placed on, it's very hard to remove. In order to remove it, they have to spray with CSF abundantly so that it floats again. So there's a lot of surface tension between the electrode and the surface of the brain. And that's in part due to a lot of line tension of the perfusion holes and the edges of the electrode that helps also the thinness of the electrode to confirm. Now, in the semi-chronic or chronic setting, we'll have to do a bioresorbable material on top of the electrode temporarily so that we guarantee that it doesn't shift with time. We have not done that yet, but we hope to do that moving forward.
Yeah, and sort of like a follow-up on this, since the contact is really strong, do you also introduce additional like holes for the tissue breathing or
To grow on top and fix it in place, right? So we do hope, yeah, that there will be tissue scarring that will fix it in place. And then the other side would be, okay, well, if there is tissue on top, how can you remove it? Because we need to remove it. And this is usually a thin layer of tissue that I'm told by my colleagues would be easy to remove, including colleagues at Neuralink.
Testing, testing. Before we get to Jonas, I want to see, does anyone have a question on Zoom? They do, we'll hear it, I believe.
They should hear us, yes.
Okay. We'll go to, last but not least, Jonas. A fun fact, he was on Jonas's PhD committee.
Yes, the good one.
Thank you for the talk.
Of course.
For a practical standpoint in terms of fabrication, like, as you go up in a number of channels, like 4096, how does your grad student or postdoc actually examine every single electrode when they're fabricating it?
I forced them to. No, they actually, of course, measure the electrochemical impedance, right? To be able to check, what is a reasonable. And then for those that are not good, they go and try to track the leads and find out what's the problem. But that rarely happens. We don't have to look at all of the leads. We have an acceptance criteria of 90% yield. And if, for many runs, our yield is below 90%, then we sit on the microscope together and we check those leads.
Oh, okay. So you don't actually optically check after every single process, right?
No,
You just
you
do it
don't.
at the end.
Yeah.
Okay, now it makes
in
sense.
the development, you do that, but not after the fact,
Okay, thank you.
of course.
Testing, testing. Actually, yeah, so we can wrap up now, but I just make a comment. I thought that this gentleman's question about why do you look at the wave analyses, I thought might be tied to Jonas's question in that when you start to sample from thousands of things, some of the random locations are going to fail. But if fundamentally the aspects of brain processes you're looking at involve spatial propagation, then you're probably more likely to be able to deal with random locations being wrong. So I just thought that was almost like a clever use of the fact that I'm recording from thousands of things. Some of them will fail. I use the appropriate type of neuroscience to get at the question. So yeah, just a comment.
Yeah.
Well, let's thank Shadi again for a great talk. Let's feed him.